everyone, I would like to present to you our study on the interaction between human interferon gamma and heparan sulfate oligosaccharides. Interferon gamma is a crucial immunosignaling molecule with various activities including antiviral, antibacterial and immunomodulatory. It regulates over 200 genes and is clinically used for the treatment and management of various diseases. However, its activity is also associated with numerous autoimmune diseases, neurodegenerative diseases and some cancers, and people are trying to find a way to block its activity, but this requires detailed understanding of its interactions and binding. Interferon gamma, shown here in red, is a homodimer which binds to two identical cell surface receptors, here in green. The binding interfaces are shown in orange and blue bubbles. This is the crystallographic signaling complex, but it lacks the positions for the highly flexible and positively charged C-terminal tails of interferon gamma, which are known to drastically change the binding affinity of interferon gamma to its cell surface receptor. So we modeled their structure, we reconstructed them and folded them using long-scale molecular dynamics simulations. They are shown here in blue, and this is the input structure that we used for interferon gamma in our studies. It is known that interferon gamma interacts with the glycosaminoglycans heparin and heparan sulfate. These are polymeric chains of uronic acid and glucosamine, which are highly sulfated and thus have high negative charge density. We studied this interaction in two different cases. The first one is the free interferon gamma in the bloodstream, and the second one is interferon gamma near the its cellular receptor at the cell surface of the target cells. In the first case, we used this hexasaccharide as a model structure of low molecular weight heparin in the bloodstream. We placed four such chains in the vicinity of the C-terminus of interferon gamma, and we found that all four chains bind very quickly to the interferon gamma molecule. This is the number of contacts between the oligosaccharides and the cytokine. As you can see, here the fourth chain associates to the globule of the cytokine, and the number of contacts saturates. Contacts are primarily located in the C-terminal tails of the two interferon gamma monomers, here and here, and also some positively charged domains, solvent exposed domains of the globule. This complex of the four hexasaccharides and interferon gamma is highly negatively charged and is thus not able to bind to the negatively charged cell surface receptors of interferon gamma. In the other scenario, in the other study, we wanted to find how heparan sulfate oligosaccharides affect the binding of interferon gamma to its cell surface receptors near the cellular membrane. So we have four different scenarios. This is the reference simulation starting from the crystallographic structure. In the next scenario, we have this full-length human interferon gamma molecule distanced at about 4 to 5 nanometers from the cellular receptors, and then we took this configuration and we put hexa or octasaccharides between the receptors to mimic the presence of heparan sulfate proteoglycans at the cell surface. We monitor the distance between the binding sites of in the receptors and the cytokine. In the reference simulations, these distances have a value of about 7 angstroms, shown here in black and brown. As you can see, when we don't have glycosaminoglycans, the positively charged C-termini binds to negatively charged domains at the receptor surfaces and prevent the globule from interacting with the receptor subunits. When we place hexasaccharide between the receptors, they interact with the C-termini and pull down the whole globule between the receptors, and one of the binding interfaces forms very well, but the other one is deformed and does not form properly, as you can see here on the distance plots. If we place octosaccharides, which are known experimentally to bind with the highest affinity to interferon gamma, they have higher negative charge than the hexasaccharides, and they very efficiently pull the globule between the receptors, and the proper signaling complex is formed. We validated our computational results experimentally. We incubated wish cells with sodium chloride, which inhibits sulfation of heparan-sulfate proteoglycans on the cell surface, and we found that the activity of interferon gamma in cells which were incubated with sodium chloride is 86% reduced compared to untreated cells. So in conclusion, glycosaminoglycans, heparin and heparan sulfate play a dual role in the interaction of interferon gamma with its receptor. On the one side, heparin in the bloodstream binds to the free human interferon gamma and prevents further binding of this molecule to its cell surface receptors. But on the other hand, we found that heparan sulfate oligosaccharides are necessary to be present in the vicinity of the receptor so that the cytokine receptor complex can be formed, and they serve as a type of co-receptor for the cytokine. I would like to acknowledge financial support by the Bulgarian National Science Fund under this grant and access to these computational resources, and I would also like to thank you for your attention. So thank you.